Look, welcome to the 2016 Peter Goodenough lecture. And I'd just like to start by acknowledging the traditional owners of this land, where we are today, the Turrbal and the Jagera people, and pay my respects to the elders past and present, and those who are around today and those to come. And a special welcome to Professor Amar Al-Chalabi, who's the lecturer for today, and he'll be delivering his lecture, What Causes MND, Motor Neuron Disease, and often called ALS, or amyotrophic lateral sclerosis in the Northern Hemisphere, or at least the United States anyway. And welcome to his wife, and there's two boys who are going to ask questions at the end, I'm sure. And Amar is Professor of Neurology and Complex Disease Genetics at King's College London. So tonight we'll be talking about what causes motor neuron disease. And look, it's a pleasure to have you all here, and particularly pleasure to welcome many people from the community and the carers and people who are suffering from motor neuron disease. And you know, it's incredible that you've come up here to listen to Amar today. Many donors, including Mr. Paul Oles, who's the CEO of MND and Me Foundation, who supports the Scott Sullivan Fellowship. And this is a fellowship at the QBI, joint with School of Biomedical Sciences, to support someone working in the area of MND. Special welcome to Clive Jackson, who's the joint trustee and executor of the Peter Goodenough estate, and his wife, Helen. Welcome. Distinguished colleagues, ladies and gentlemen. So Peter Goodenough was born in Cornwall, United Kingdom in 1936. And he was born into a farming family. This was a complex farming family which did a bit of everything, really. And having lived through the Second World War, he could remain connected with the family in very different ways, but he did a fair bit of traveling and study, and he actually graduated with a degree in agriculture and piggery, as might say. And he migrated to Australia in December 1964, sailing with the Castel Felice. And he came here under the Assisted Passage Migration Scheme. For those of you who might know this was people who were assisted to migrate to Australia to build the population here, and it cost them £10 to come to Australia, very different to now. And they were called the 10-pound POMs. So, you know, this is the Australian vernacular. POMs. I guess we don't call people POMs anymore, but certainly, when I came here in the early 70s, there certainly were POMs and WOGs around then. So that's a big change. So Peter Goodenough succumbed to the devastating disease, motor neuron disease, and he passed away in 2004. But because of his personal battle with the disease, you know, he had an inspirational bequest, and he bequested the money to the University of Queensland, to the Queensland Brain Institute, to form the Peter Goodenough and Wantox Laboratory. So the Wantox Laboratory, the Wantox were his dogs, and he initially bequeathed $3 million, which has grown to about $10 million over the years, and it's one of the largest bequests to the University of Queensland. So it's really probably the second biggest donation to the University of Queensland after the main bequest. And for those of you who read the story of the main bequest, probably under some better circumstances than the main bequest. To tell you, if you haven't read the book, you should read the book about the main bequest. Very interesting and challenging story. So Wantox means friends, and these were his dogs who supported him and, you know, kept him sane over the many years in Papua New Guinea. So Peter Goodenough actually came to Sydney and pretty much got on a train and moved to Queensland. And here he set up a very large civil engineering company with interests in North Queensland and in New Guinea. And that's pretty much where he made his money and bequeathed the profits from that. So at the time of the bequest, in discussions with the estate trustees, and that was Perry Bartlett at the time, who was the founding director here, it was decided to start this lecture, the Peter Goodenough Memorial Lecture. And the inaugural lecture was presented by Sir Gus Nossel in 2008. Since then, the speakers have included Professor Harvey Whiteford, Sarah Caddick from the Gatsby Estate, Chris Shaw, also from his college, and Professor Pat Sullivan, and last year was Peter Beattie, a past Premier of Queensland. So it's great pleasure to welcome Amar here to give this lecture. Amar graduated in medicine from Leicester University and trained in neurology. And soon afterwards completed a PhD in motor neuron disease and is currently a consultant neurologist and in London and director of the King's MND Care and Research Centre, an MND Association endorsed Centre of Excellence. So from very early days, Amar has been a leader in genetics of MND and received many awards and prizes, starting with the work he did with his PhD, for which he received the MND Association Charcot Young Investigator Award. And last year, or this year I should say, the Sheila Essie Award in ALS from American Academy of Neurology this year. So his research focuses on understanding the causes of MND and the clinical management of MND. He's a passionate clinician. For those of you who've heard him talk on YouTube, on the videos on YouTube, he's a great scientist and a passionate clinician. So he has published over 200 research papers, and his team have identified or helped identify most of the known gene variants in MND. And you know, the way MND is headed, probably the work of people like him are going to lead to possible cures for this disorder in the future. He leads the European Consortium Strength using genetics, population registers, and environmental questions to identify subtypes of MND. It's recently showed that on average 6 different molecular steps are involved in MND, and they're thinking about really the biochemical mechanisms that might underpin this disease. I'm sure you're looking forward to his lecture. Welcome, Amar.
Thank you very much, Pankaj, for introducing me. And thank you very much to the Queensland Brain Institute for inviting me, to Naomi Ray for organising it, and to the Peter Goodenough Trust for funding this lecture. Also, I'd like to thank and acknowledge my team in the lab in the UK, because most of what you're going to see today has been done by them. So the theme of my lecture is risk factors and motor neurone disease. I may sometimes call motor neurone disease ALS during the lecture. For me, they're synonymous. And the theme is nature, nurture, age, and chance. This is not Voldemort. It's actually my head reconstructed in 3D from an MRI scan. And if I chop away a bit of my head, you can see the brain underneath and the motor cortex, which is lit up there. This is where the motor nerves live. And the cell you can see there is an upper motor neuron. And it sends its axon, which is basically a long wire, down either to the brainstem or to the spinal cord, where it talks to another neuron, which is called the lower motor neuron, and that sends its axon out to the muscle. And human beings are unique in having such a simple circuit. You've just got these 2 nerves, and because of that, we have a complete mapping between the neurons in the cortex and what happens in the hand or the leg. And that's what allows us to be able to make watches or build a space shuttle or do gymnastics at Olympic level. It's what gives us such an advanced motor system. But that also is the Achilles heel of the nervous system, because a bit like having a thoroughbred racehorse or a fantastic sports car, if you've got a very high performance system, it's much more difficult to maintain. And when it goes wrong, it's much more expensive and difficult to repair compared to a simple system. So an animal like a mouse has lots of nerves in this chain, and if something goes wrong, they can just grow the nerve around it and fix it, whereas we can't really do that. But the mouse then can't have the fine motor control that we do. So it's a two-edged sword having such a good motor system. So as I said, this is the upper motor neuron, the one in red, and the one in yellow is called the lower motor neuron. And if you damage the upper motor neuron for any reason anywhere in its course, then the person will develop weakness because they can't get the signal to the muscle. They'll develop spasticity, which means that the limb becomes very stiff, and they develop brisk reflexes. So when we tap a tendon hammer in the clinic, the person's limb will jerk very quickly. If you damage the lower motor neuron for any reason, then again there's weakness because the circuit is broken. But in this case, what happens is muscle wasting, because the lower motor neuron normally sends signals to the muscles to keep them big. And a thing called fasciculation happens, which is like a bag of worms under the skin, or twitching of the muscles. And there's loss of reflexes. So as a clinician, if we see this strange combination of wasting with brisk reflexes, which should never occur, or some similar combination of lower and upper motor neuron signs, then motor neuron disease is very high up on our list of possible diagnoses. This is what it looks like for people in the audience who don't know. This is someone whose hands have been affected. And what you can see is wasting of the muscle under the thumb and under the little finger. And the fingers are clawed as well. And that's because the flexors in the arm are always stronger than the extensors. It's easier to bend your hand than to straighten it. And when they both become weak, then the imbalance is more pronounced, and so you get flexion of the fingers. And if you look at the back of the hand, then you can see there's guttering of the muscles there, and particularly the one between the thumb and the forefinger, which is called the first dorsal interosseous. This is the spinal cord of somebody with motor neuron disease. So the front of the spinal cord is at the top, and the back of the spinal cord is at the bottom here. And what you can see is this area I've circled, which is a very pale area. That's the area where those upper motor neurons send their wires down to talk to the lower motor neurons. It's called the lateral corticospinal tract. And you can see that it's paler here than it should be. It's paler than the surrounding tissue. And that's because it's lost myelin as the nerves have been damaged. And then if we zoom into the other part, I'll just see if this laser pointer works. Should be that. So this part here is called the anterior horn. And that's where the lower motor neurons live that those upper motor neurons are talking to. If we zoom in on that, it looks a bit like this. So here you've got a normal motor neuron. So the whole field should be packed with those normal motor neurons. In fact, it's not. And you can see these shrunken, dying motor neurons there. And if you're very sharp-eyed, you might notice that even in this normal motor neuron, there's a yellow blob there. Can you see that? That's an inclusion. And I'll show you an inclusion properly now. So this is an inclusion. It's a blob of protein that's stuck in the cell. And we don't know what these blobs mean. We don't know if they're toxic to the cell, or if they're simply like a gravestone saying that the cell is damaged, or if they're just a bystander that just happens to be there. But we see them in people with motor neuron disease, and they're specific. This particular type of inclusion is specific. And they contain a protein called ubiquitin, which the body uses to tag damaged proteins so they can be destroyed. And the other characteristic feature we see is the neurofilament accumulation, which is what you can see there. It's a big blob of the cell skeleton proteins that have just dismantled and become a big blob instead of being a nice structure. Now, you'll hear lots of different types of motor neuron disease. One of the first things people say to me in clinic is, what type of motor neuron disease do I have? And if you ask different neurologists, they all have their own particular classification. There's no easy way of saying, well, you've got this type or that type. This is my particular grouping that I use when I'm describing patterns of motor neuron disease. But we don't know that those are meaningful classifications. They're just based on my clinical experience. So one thing we tried to do was ask a machine to look for patterns in the clinical data and say, are there actually types of motor neuron disease that a machine could see objectively? Are there different patterns that really do exist objectively? So we used a method called latent class cluster analysis, and it pulled out what you can see down there, which is 5 different groups. I'll show you those in a moment. Now, if we take the patterns of motor neuron disease that I see in clinic, then you get, you can plot survival of the people with motor neuron disease of those different types. And what you can see here is called a Kaplan-Meier curve. The way it works is, at the beginning, 100% of people are alive, and every time, as time goes on, the graph moves along. This is time on the x-axis. And every time someone dies, the graph moves down a bit, down to the correct proportion left. So what that means is, if a graph is more to the right or more horizontal, then the survival is better than if it's to the left or dropping vertically. And you can see that people with the black line here, which is bulbar palsy, so that's people who have swallowing difficulty, they have particularly poor survival. So it's the leftmost line. Whereas the people here with the purple line, they have a pattern called flail leg syndrome, which is a floppy weakness in the legs. They have much better survival. So the patterns we see in clinic do correspond to differences in outcome. But actually, if we use the machine classification, it's much, much better than a human being. So it finds these two groups a bit like these two groups, but it also finds these people here who have 10-year survival and 15-year survival average. And it also finds this group of people here who never die. And this is using information that we only have on the very first visit in clinic. So that means there are hidden subtypes of motor neuron disease that we're not spotting as clinicians, but are there actually to find if we knew how to find them. This is the model we use as researchers. So we're conceived, we're born, at some point we develop disease, that is the end disease we're going to get, and we die. And we only see people after their disease has started because we have to diagnose the disease. So we observe what happens between disease and death as the clinical features. And we use that to guess what happened before the person developed disease, because we don't know what happened before that, because we haven't seen them before that. And then we use that to try and make a treatment that acts after we see them. So this is the only way currently for a relatively rare disease like motor neuron disease to develop a new treatment. You have to use this kind of convoluted loop of logic. So understanding what happened to cause the motor neuron disease is important for designing a treatment. So that's where the nature, nurture, age and chance comes in. So to illustrate what I mean by nature, nurture, age, and chance, I'm going to use the Great Fire of London. This is what London looked like on September the 1st, 1966. So actually, for people who've been to London, this is St. Paul's Cathedral. And you know now it's got a dome. That's because that was built after the Great Fire of London. So this was 1st of September 1666, and this was the 2nd of September 1666. A huge fire swept through London. And if you look at a map of the city after the fire, this is what it looks like. And I've just ringed here the area that's been burnt. So everything in that area you can see is much less populated than everything else. So if you look around these areas around here, there's lots and lots of houses on the map, and here there's hardly anything. The fire started in this part of London, in Pudding Lane, in a baker's shop. And the buildings that survived are those in red. They're the only ones that made it through the fire. So we could use this as a model to try and understand disease, because why did some buildings survive, why did others not? So you can imagine the ones that didn't, it's the equivalent of having motor neuron disease. The ones that did survive is the equivalent of not having motor neuron disease. So nature, by which I mean what is the composition of the building, all except one wooden structure burnt down. So being wood, if you're a wooden building, you're susceptible to fire. If you're a stone building, most of those survive the fire. The wooden buildings were close together. So that's not part of their structure, that's to do with their environment. That's how they happen to be. If you'd built them separately, then there might have been a fire break and the fire wouldn't necessarily have spread so fast. Whereas the stone buildings were isolated and they were surrounded by walls. So even if there was flammable things in those buildings, it wasn't able to catch fire because the fire just didn't get to them. So that's the environmental part, that's nurture. Age also played a part because older buildings were less resilient to the fire than newer buildings. And there was an element of chance because, for example, the building you can see there in blue, just at the edge here, that was just outside the fire zone, just by chance. It's a wooden structure, it's got all the risk factors. It's a wooden structure surrounded by other buildings, but it didn't burn down. And in fact, you can still go to it. It's the pub, the George, on the Strand. So we can't really discuss chance in a research setting. We can't really identify factors that are chance-related. It's very difficult to study them. So we have to discard that part of the nature, nurture, age, and chance. But we can study age. So the peak age of onset of motor neuron disease is about 60 to 70. It depends whether you're looking at patients who come to clinic or in population registers. But if you look at the lifetime risk, then it looks like this. So the graph I showed you just now was just the number of people at each age group who get it. But this is actually your risk of developing motor neuron disease at each age group, your cumulative risk. So you can see by the time you've reached 85 or 90 up here, you've got about a 1 in 300 risk of developing motor neuron disease. So that means if you're on an airplane, on a long-haul flight, maybe 2 people in that flight will develop motor neuron disease. It means 1 in every 300 people will die of motor neuron disease. So that's much higher than you might think when you say the incidence is 1 to 2 per 100,000 person years. You may have heard those kind of numbers. And those numbers are comparable, in the UK at least, to the incidence of multiple sclerosis, the lifetime risk of multiple sclerosis. The lifestyle and past exposures aspect of it, the nurture part, is also difficult to study. And the reason it's difficult to study is because there are effectively infinite numbers of things that you could be exposed to at any point in your life that might cause it. For example, if apple pips that you ate when you were 3 years old cause motor neuron disease, we would never detect that in any kind of questionnaire. So the search space is very, very large for environmental studies, and that makes them very difficult to do. But there are a few interesting results. So this is the geographic distribution of everybody with motor neuron disease in a 10-year period in the southeast of England. The black dotted line is just the boundary, the westward boundary. The north boundary is the River Thames, and then the east and west boundaries are the sea. And you may see some clusters there. If you look, so that there are some areas where there seem to be little clusters of motor neuron disease. I've circled the ones that I see. So I see clusters around the coast. I see some going along the motorways, and a big cluster in London. But then, if you think there are more people who retire by the coast with motor neuron disease in the UK, because it's a nice place to retire, and we know that age is a risk factor for motor neuron disease, so it's not surprising that there's more motor neuron disease by the coast. It's simply a factor of people being older. And more people live in London, and so simply by weight of numbers, you're going to have more cases in London. So they're not real clusters. So when you're doing a clustering study, people often come to me and say things like, oh, 6 people in my road have developed motor neuron disease, or 3 people who went to this school. It's very difficult to know if that's a real cluster or not because of these kinds of effects. But there are ways to try and balance it. So this is, if we zoom in on the London part, the areas in black do have an increased risk. It's about double the background risk. And the areas in white have a very reduced risk compared to the other areas. And our hospital is here. And you can see it's next to an area with high risk and next to an area with low risk. So it's not that it's an ascertainment thing. These are real, or probably real, areas of increased or decreased risk. And lots of studies have looked at clustering, and this is a study from Finland, a very nice study actually. It looked at people at birth, their place at birth and their place at death. And that shows these 2 clusters in Finland. A study in Italy also shows clustering. This is a heat map showing where the greatest number of cases are. And every study that's looked for clusters has found them. But we still can't go from finding the clusters to knowing why there are clusters. Another risk factor you may have heard of is exercise. So if you speak to most neurologists, they will say, oh, lots of my patients are very fit. I often have people saying things like, oh, it was when I was running a marathon that I developed a foot drop, or when I was lifting weights in the gym, I couldn't just lift that last weight. So we do hear that a lot. And so people thought, well, perhaps exercise is a risk factor. But actually, you can't say that just from seeing people in clinic because, for example, if exercise is actually protective and slows the disease course down, and not doing exercise makes the disease go quicker, then all the people who don't exercise will die before they get to us in clinic because it takes about a year to be diagnosed for most people. And so the people we see in clinic will be the ones who are left who are the ones who exercise. So it would be a bias. So it's very difficult again to study whether exercise is important. Gulf War veterans, deployment to the Gulf War seems to be a replicable risk factor. And other studies of exercise have been inconclusive. So one of them you may have heard is football. So this was a paper that came out in Brain. It happened because Italian football players were thought by an Italian prosecutor to drink a lot of alcohol and take a lot of drugs, and they wanted to investigate that. So they said, let's do a study to see what these footballers are really dying of. And there seemed to be an excess of people with motor neuron disease in the footballers. So the study was done again in a proper systematic way. And out of 7,325 people in the study cohort, they found 5 people with motor neuron disease. And according to the statistics that group used, they would have expected less than 1 case in that cohort. So that means that the risk of death was about 6.5 times. The risk of death from motor neuron disease was 6.5 times what you'd expect. But actually, if you do the analysis a different way, looking at lifetime risk and taking into account the age structure, then you find there's no increased risk. So again, it's controversial whether football is important. Here's another example of how football this is another example of clustering with a condition. So these are 3 people we saw. They're friends. All of them played football. They all developed motor neuron disease at roughly the same time. They grew up in the same small village in Kent, which is a county in the southeast of England. They all played amateur football several times a week for up to 28 years. 2 of them played on the same team, and the 3rd one was on another team that they frequently played, and they all played on the same pitches. They all developed MND at the same time. 2 of them died on the same weekend. In fact, a 4th person from that team has also been affected since. But they all shared other potential risk factors, so they were all electricians, and they'd all received electric shocks as part of their work. They all smoked heavily. They'd all broken bones. So it's not just football necessarily that is the risk factor here. They all went to the same schools, they drank the same water because they lived in the same villages. It's very tempting to draw the target after you've got the arrows, but you have to be really careful about that. So if smoking is a risk factor, then he's in trouble. The best quality studies do show smoking is probably an increased risk, and there's probably a dose response. And there's a Dutch study that suggests it also worsens survival. But our clinic population, certainly in the UK, don't have many smokers. And you can show that that's because the people who smoke are depleted from the population by dying before they see us. So that's because it worsens survival. And there's some studies that show that the risk is greater for women who smoke than for men who smoke. So if we look at nurture and motor neuron disease, so these environmental risk factors, just to summarize, it's difficult to draw firm conclusions. There are geographical clusters, it's not clear why. Smoking is probably a risk factor, exercise might be. So the only thing left really that we can draw any conclusions from potentially is nature, is the genetics. So I know many people in the audience are not geneticists, so I'm just going to explain a bit about genetics. This is DNA. So this is the instructions in each of your cells to make you. And the DNA you've got in each cell is about 1 meter long. If you unraveled it and made it into a ladder, then it would reach to the moon and back twice from each parent. So it actually moon and back twice, 4 times. And it's made of 4 different types of teeth, which we label GATC. So just like you've got incisors, canines, premolars, and molars, there are 4 types of teeth in DNA, G, A, T, and C. And the exact order of those teeth is what determines the proteins that are made and how you develop. Now, if you have, it's 3 billion teeth long, each strand of DNA, each set of DNA. If you have that much, it's going to be all tangled up. So your body tries to store it in a neat way. For people in the audience who are old enough to remember telephones with a wire, you know, they all get knotted up very easily. So your body has this system which are called histones, which are these blue things with the little wires you can see. And they self-assemble and they wrap the DNA around themselves. And then the coils coil, and then those coiled coils coil more. So you can see that happening now. This all happens spontaneously. These are all self-assembling. And then those coiled coils coil even more. And then those coil even further. So the whole thing's got a package into a microscopic cell, even though it's long enough to be 1 meter long if you unraveled it. And then those things are packaged into these things called chromosomes. And the chromosomes are in the nucleus of the cell. So every time a cell divides, you've got to copy all of that DNA. And you mustn't make a mistake in doing it. So your body has a machine that does that. This is the machine. It's called Taq polymerase. It's called polymerase, DNA polymerase. And you can see, if you follow the DNA downwards, you can see that it's 2 strands. The 2 strands are unzipped, and one of the strands can be read forward, and that can be replicated easily. So that's the strand you can see here going off to the right. The strand on the left, the other strand, is being read backwards. So it can't be copied easily biochemically. It has to be looped, copied backwards, and then unlooped. I can look at this lots and lots of times and never quite understand it. But this is real time. And this is happening all the time in your body. You make, just for example, for red cells, you make 1 million red cells a second. So all of your cells are replicating that kind of fast. So you're having to do this a lot in your body all the time, and you mustn't make a single mistake. Because if you make a mistake, the mistake corresponds to a mutation, the mutation corresponds to a change in your protein, and that could be dangerous. So our job as geneticists is to find in that ladder that goes from the moon to the moon and back twice, the single rung that is wrong that might be relevant for motor neuron disease. So that's quite a difficult task. But the first question to ask really is whether there's a genetic component to motor neuron disease at all, because there might not be, in which case it's not worth looking. So one way you can answer that question is by looking at twins. If you're an identical twin, then you share all of your DNA with your twin. And for the most part, you share all of your environment, at least while you're growing up. If you're a non-identical twin, then you only share 50% of your DNA because you're a sibling. But again, you share the same amount of environment as if you're an identical twin, generally. So twin studies are quite a good way of looking to see whether a condition is genetic or not, because if it's genetic, you'd expect the twins that are identical, who share more of their DNA, to both be affected by the condition more frequently than if they were non-identical. So if you look in twins for motor neuron disease, you can see that when the twins are identical, out of 49 twin pairs, in 5 cases where at least one was affected, in 5 of those cases, both the twins were affected. In 44, only one was. Whereas if you look at the non-identical twins, on no occasion were the non-identical twins both affected with motor neuron disease, only one was. And if you crunch the numbers, that tells you that the heritability of motor neuron disease is about 60%. So that means that the variation that's contributing to whether you develop motor neuron disease or not from genes is about 60%. So 60% of the variation in your genes is responsible for motor neuron disease. Conversely, it suggests that about 40% should be environmental. Another way we can ask whether it's genetic is to ask whether there's an increased risk to relatives if somebody has motor neuron disease but no other family history. So if you have one person who's been affected in your family, is there an increased risk for you or for your siblings? So we approached that in our clinic. We looked at all the people who'd been in our clinic for the last 15 years who didn't have a family history when they saw us, and we chased up whether subsequently one of their family was affected. And when we, again, crunched the numbers, we found that although there was an increased risk, in technical terms there's an increased risk, but in practical terms it's irrelevant. So the risk of not developing motor neuron disease drops from about 99.7% to about 98%. So people, even though there is technically an increased risk, people with a family history of motor neuron disease will overwhelmingly die of heart disease, stroke, or cancer instead of motor neuron disease. But from a scientific point of view, it's important to know because it means that there is a genetic component, probably. The other thing we know is that even if there isn't a family history, the family size is important, and you can think about this quite easily. If you had 100 siblings and you had a disease gene, that increases your risk of getting a disease. It means you've got a 50-50 chance of getting the disease. So if your parent has the disease gene, you've got a 50-50 chance of inheriting it, and then a 50-50 chance of manifesting the disease. So if you have 100 siblings and you're the only person in the family affected, 50% of your siblings, that's 50 of them, will have carried the gene, and 50% of those, that's 25, would be affected. So you'd have 25 siblings who are affected. It would definitely look familial. On the other hand, if you only have one sibling, it's quite likely that one sibling wouldn't be affected. So your family history, your family size, sorry, affects whether you get a family history or not. So just saying somebody has a family history of motor neuron disease doesn't necessarily mean that people who don't have a family history don't have a genetic contribution. The other way we can see this is like this. This is a pedigree of a family with motor neuron disease. So the way pedigrees are drawn is males are squares and females are circles. And each generation comes down as a stalk. So we've circularized this pedigree. Normally they'd be horizontal. And so this is the first generation, second generation, third, fourth, fifth, sixth. And everyone who's affected is shaded in black. And what you can see is, for example, these two people here are fifth cousins. So if this person came to my clinic, they wouldn't know their fifth cousin. I'm sure none of you know your fifth cousins. So they would seem to have sporadic motor neuron disease, no family history. But if you look at this person here, if this person came to my clinic, then I'd say, well, their father was affected, and actually they've got another family history in a relative. So this looks like familial motor neuron disease. I can tell you in this family, there is a single gene that causes motor neuron disease. But you can also see the risk in this family is about 1 in 10. I told you the background risk is about 1 in 300. If it was a gene that really greatly increased your risk, for example, a gene that might be like Huntington's disease genes, then you'd expect about 50% of the risk people to be at risk, not 1 in 10. So you can see there's plenty of people here who should have been affected who weren't. So this person who's transmitted it to these two people isn't affected. And the same here, all of these people must have carried the disease gene but weren't affected. So having a disease gene on its own isn't enough to cause motor neuron disease. Because the genetics of motor neuron disease is advancing really rapidly, there is a website you can go to if you're interested, which is ALS online database, which is updated continuously and has tools for researchers and a few for interested lay people. So a mutation is where a single tooth of the DNA, a single base pair, is altered, is misread. And that matters because normally the different cell machines lock together, as you saw in those animations. The machines really have to work together very tightly. So you can imagine here, for example, this is one protein, that's another protein. If this protein had a misprint in how it was designed in the DNA, then it might be misshapen like this and have two blobs there so that the other thing doesn't fit properly, and then the machine won't work properly. So mutations are important biologically, and they often cluster in one part of a protein. So that's why we look for mutations in disease. This is the state of motor neuron disease genetics at the moment. On this graph, you've got the year of discovery, gene discovery on the x-axis, and you've got the cumulative count on the y-axis. And the size of each sphere is a measure of how many publications mention motor neuron disease for that particular gene. So it's an indication of how important the research community thinks that gene is. And you can see that the gene discovery is doubling every 4 years. So that's a really dramatic increase in our knowledge of motor neuron disease. Now, you might argue, well, this is the latest gene here, and therefore it's below the line. But actually, 2 weeks ago, 2 papers came out in Nature Genetics showing another 4 genes. So actually, the count goes back up onto the line. And it means that over the next 4 years, we can expect another 35 motor neuron disease genes to be found. And each of those genes gives us another piece of the jigsaw puzzle to help us understand why the disease happens. So why, how are these gene discoveries happening? Why couldn't we discover this many before? And the answer is multiple things. One is better technology. The kind of technology we used in the past was very, very laborious, very time-consuming compared to now. We can now sequence a whole genome of somebody in a week. You can do panels of people very, very quickly for about $1,000. The whole genome sequencing project that began in 1984 took 16 years to sequence one person's genome. So and it cost £150 million just for the consumables. So we can now do that in about a week for $1,000. The other reason is bigger sample sizes. All the researchers across the world collaborate very strongly together now. So the sample size in thousands is roughly corresponding to the gene count. So the latest study had 40,000 participants, and we've now got about 40 genes. Another reason is that we've got harmonized sample sets. So what I mean by that is researchers in Queensland who are collecting DNA do it the same way as researchers in the UK or researchers in the US. And so we can combine all of our data in a meaningful way. And we've got a better understanding of clinical features, so we know what to call motor neuron disease and who to include in the studies. This is an example of a current study called Project Mine. It's an international whole genome sequencing study. And the aim is to sequence 15,000 genomes of people with motor neuron disease and 7,500 people without, and then compare them to see which variants might be causing risk. And the first few thousand sequences are now being analyzed. We've got multiple layers of clinical data from each person who's participated in that study, from every research group. It's a huge international study. And all of the data collection is being done in a harmonized way. And it means that we can share the data, and all the researchers want to share the data, which is really important as well. But to put in perspective how much data this is, recently Google very proudly announced, if you've got an Android phone, you'll know that your photos back up automatically to the Google Cloud. And for example, I not only have my photos backing up, but all my previous photos I've ever had digitally, I've uploaded to Google as well, and all your video. And so they announced that the entire world's photo and video uploads that have gone up to Google so far is 13.5 petabytes. That's 13,500 terabytes of data. Now, each genome is 1 terabyte roughly, so that's a big hard drive. So the Project Mine expected output from that international genome sequencing study is 22.5 petabytes, 22,500 terabytes. So it's a really huge amount that even dwarfs all of the videos and photos that have been uploaded to Google so far. So just handling that kind of data, how do you even plug in? You can't plug in 22,500 hard drives into a computer, obviously. This is just to show the effective sample size. So this is the signal of a gene we know is very important for motor neuron disease. It accounts for about between 7% and 10% of all people who have motor neuron disease. This was the first proper signal we saw for the gene. You can see it's there, it's the red signal. But this is the latest study. So this first study is 12,000 people. This is with 40,000 people. You can see it's an unmistakable skyscraper. And having these larger samples also means we can discover new genes. So we confirmed these disease genes, but we also found some new motor neuron disease genes using this technique. I'm not going to go through a massive list of genes, but I do want to use just a couple to illustrate things. So SOD1 was the first motor neuron disease gene to be identified. It was identified in about 1994. In fact, it was the first disease gene, one of the first disease genes for any disease. So cystic fibrosis, Huntington's disease, and SOD1 genes were the first 3 found. And we know it's mutated in about 25% of people who have a family history, and in about 3% of people who don't have a family history. But although we know there's about 150 different mutations in this gene that can all apparently cause motor neuron disease, we still don't know why even now, since 1994, so that's 22 years later, we still don't know how they cause motor neuron disease. So that's one important thing to know. Just because you find the disease gene doesn't necessarily mean that you will discover the ultimate cause, but often we can find it. So this is another disease gene. This is called TDP43. And remember those inclusions I showed you at the beginning? So we now know those inclusions contain TDP43. And that's a link from the pathology to the genes. And TDP43 is involved in a thing called RNA processing. It splices together RNA. So I'm going to show you what RNA processing is. So this is the DNA strand that you saw earlier. And what you can see there is a little machine that's going to read the gene. So this happens in all of your genes. It's going to read the gene and convert it from DNA into RNA. And then that RNA is later converted into a protein. So here it goes. So the yellow strand you can see shooting up is the RNA being made. All those little yellow things you can see are the nucleotides that go to make the RNA that are just floating around. This is, again, roughly real time. So that shoots along. Eventually, it'll come to a stop code on a little sequence that says stop. And when it comes to that, it drops off. That's just a zoomed-in view. So you can see it's very, very fast as a machine to do this. Okay. And eventually, once it's dropped off, it floats out. So there's a lot of other things that go on here, so it's spliced together. But eventually, it comes out, and it's connected to a thing called a ribosome. And the ribosome is a machine that converts that RNA sequence into a protein. And the protein is the little machine that, when lots of machines are put together, they make you. So these green things are called tRNAs, and each one carries, it's like a little key. Each tRNA recognizes a specific three-tooth sequence, and at the other end of the tRNA is an amino acid that's specific to that sequence. And so you can see it chugging in there. The tRNA is clipping on, and a little amino acid is being joined onto the protein chain. So if you cut away, you can see there's the protein chain being made along the top. So eventually that protein will fold up and make a new molecular machine. So if there's a mistake in this sequence and that molecular machine is made wrongly, and then you get disease. But the real problem is there's many motor neuron disease genes involved in RNA processing, we now know. In fact, there's something like 10 motor neuron disease genes that are involved in this pathway. But RNA processing happens in every single cell all the time, so it's not clear why they should cause motor neuron disease. Here's another clue from genetics. Ubiquitin is the protein that I told you tags these damaged proteins. It's in these inclusions. So the inclusions contain ubiquitin. We now know that many genes that are involved with ubiquitin also cause motor neuron disease. So this is what the ubiquitin pathway does. So here the proteins are tagged with ubiquitin, and then they assemble. This is the tagging taking place. They assemble and get taken into this thing called a proteasome, which is like a big dustbin in each cell. And the dustbin does some enzymatic digestion, so it chops up all the protein back into amino acids, and then those amino acids can be recycled. So we know when this dustbin system goes wrong, then when it seems to clog up the cell, and then the cell is at risk of dying. And then the third group of genes that are important are cell skeleton genes. If you remember, I told you about the cell skeleton when it accumulates in the first part of the neuron. So lots of cell skeleton genes seem to be involved in motor neuron disease as well. And the cell skeleton looks a bit like this. So what you can see here is different proteins that have been put together to make the cell skeleton. These are actin filaments, so they're very, very thin filaments. And we might see some microtubules being made in a moment. So there's a microtubule, and this thing on the top is a little molecular machine called dynein that's carrying a vesicle, and the vesicle is a bag carrying neurotransmitters. And those neurotransmitters get taken to the end of the nerve cell and then secreted out, and then they talk to the next nerve cell or the muscle and make the contraction of the signal happen. So we've got at least 3 parts of the jigsaw. We've probably got a few more, which are RNA processing, the dustbin system of the cell, and the cytoskeleton system of the cell. And then there's a few other bits and pieces that are coming together. But hopefully, as we find these other genes, we'll be able to put more and more bits into this jigsaw and connect all the pieces and say, well, actually, that's why these 3 different parts, or that's why these 5 different parts are involved. The other important thing to know is that motor neuron disease genes can cause more than just motor neuron disease. This is a thing called pleiotropy. So what we've done here is, on the y-axis, you've got the year of discovery of a particular gene, and then every time there's a publication, the circle gets bigger. So genes that are thought to be important are bigger, and genes that are less important are smaller. And then the more that gene is involved in frontotemporal dementia, rather than motor neuron disease, the more to the right I've plotted it. So you can see this gene, C9orf72, which is very important in motor neuron disease, causes motor neuron disease and frontotemporal dementia roughly 50-50. Whereas if you've got a mutation in SOD1, it generally only causes motor neuron disease. And these other genes are somewhere in between. And then you've got this that really just causes frontotemporal dementia with a little bit of motor neuron involvement. So motor neuron disease genes can cause more than motor neuron disease, and that's a really important thing to know, because it means when we're asking a family history, we have to ask about more than just motor neuron disease. So here is the big jigsaw piece that I haven't shown you yet, which is the C9orf72 gene. So everybody has this gene in their body, and within any gene are sections called exons, which code for protein. They're the actual bits that are read by that RNA processing machinery, stitched together and then made into a protein. And between them are little flanker regions which seem to be involved in regulation of the gene and other factors. And everybody has, in their C9orf72 gene, in between exon 1A and 1B, a little sequence that reads GGGGCC twice. So it just goes GGGGCC, GGGGCC. But for many people with motor neuron disease, that repeat is expanded hundreds of times. It's really, really gigantic. And we don't know why, but there's a lot of work going to this, and we're beginning to understand why. This causes toxicity to the motor neurons, which die. And this is responsible for between 7% and 10% of people with motor neuron disease, this expansion. And it can also lead to frontotemporal dementia as well, or it can lead to both, or it can lead to nothing. So that pedigree I showed you, where only 1 in 10 people were affected, when you might have expected 1 in 2 people, have this disease mutation. So you can carry this disease gene mutation and be perfectly okay. Or you could get motor neuron disease. And these expansion mutations, they're quite common in neurological disease, and this is how they happen. So the yellow bit is one of those repeat sequences, and that polymerase, that machinery that tries to copy the DNA, doesn't like repeated sequences, and it slips. A bit like if you're old enough to remember vinyl records, the needle can slip. The same kind of thing. So you get the slip, and you get this big loop of extra DNA, and then when that's replicated again, it can be unraveled by the machinery, and then it's just copied properly, and you've got this expansion that's then transmitted to the next generation. So that's how you can get these expansions, and they can get very, very big, and then they tend to cause neurological disease. So again, we don't yet know the mechanism of action, but there's a lot of ideas about it. This is the model that geneticists use to understand diseases like motor neuron disease. It's called the liability threshold model. So we imagine that everybody in the population carries some burden of risk factors. Most people carry an average number of risk factors. Some people carry hardly any, and some people carry a lot. And if you carry too many, you pass the threshold and you develop disease. So what that means for an individual is that you're born with a genetic load, you get cell damage with time, and then you accumulate environmental exposures, and if you get enough of these risk factors piling up, then you'll develop disease, and if you don't, you won't. So if you do develop a disease, if you do develop enough of these risk factors, you'll get, in this case, motor neuron disease. And we think that some people have big genetic loads that they're born with, so those people would be people with a family history, and other people might have a small genetic load or a number of small factors. And it's really important to know, for treatment purposes and for design of, and for understanding clinical trials and things like that, whether people have just 1 or 2 big risk factors, or whether everybody has lots and lots of risk factors contributing. So I'm going to try and illustrate that. I took this picture of your Queensland sky a few days ago, and you can see in the middle there's the Milky Way here, and then there's some constellations around here. So it's a bit like thinking about that. Is motor neuron disease like the Milky Way, where you've got lots and lots of faint stars each of which contributes a little bit to the light and then you get the Milky Way? Or is it more like constellations where you have a few big stars that you can see and then they're grouped together to make a constellation? So is it a few big risk factors in each person, or is it lots and lots of little risk factors? And we can try and answer that using genetics. And we can do that by cutting up the frequency of the gene variations we see. And if we do that, one of the things we see is that rare variation explains motor neuron disease much more than common variation. And that's the opposite of things like height or schizophrenia. And that tells us that probably in each person, there will be a number of big risk factors rather than lots and lots of little risk factors that all accumulate to cause the disease. So in other words, sporadic motor neuron disease might be caused by a number of what are called private mutations. So you might be the only person with that mutation in the world. So that makes gene discovery quite difficult because you can't then say, well, that must be the risk factor because every person has hundreds of different private mutations, so you wouldn't know that it's that specific one. But if we can accumulate enough evidence for that particular gene, it means you might be able to make a personalized treatment for that person. So understanding motor neuron disease genes is important because we can understand the mechanism, we can understand how the disease happens and therefore design a treatment. We can potentially use a gene therapy for things like C9orf72 where it accounts for a large proportion of people. We can use it potentially to predict survival. If you can find genes that correlate with how long someone lives, then you might be able to also model that as a treatment. You could use it for stratifying for trials. If there's a mutation or a variation, that means you're going to have a worse outcome, and you do a clinical trial, and all those people with that variation are in one arm of your trial, it's going to make the results either look very good or very bad by accident. We could use personalized medicine because we can do gene therapies and things like that, and it could also be useful for classifying motor neuron disease. But all of this still leaves us with some key unexplained features of motor neuron disease. Firstly, there's an increasing risk with age, but people are born with these mutations. So why don't they develop disease right at the beginning? Why do you wait until you're 50, 60, 70? Why does your body then develop motor neuron disease instead of at the beginning? There doesn't seem to be a single cause of motor neuron disease. Lots of things seem to be able to cause it. We can't find environmental risk factors that are reliably replicated. One gene variant can cause multiple diseases. You saw it some disease genes can cause motor neuron disease or frontotemporal dementia, for example. You have reduced gene penetrance, so that means that even if you carry a disease gene, you won't necessarily develop the disease. It seems to start in one region and spread, but all your cells are carrying these risk factors. Why does it start in one region and then spread? Why doesn't it just affect your whole body in one go? And it's specific to motor neurons, but does include other neurons. Why is that? So one way we might be able to explain that is by taking a leaf out of cancer, the cancer world. So in cancer, we know that it seems to be a multi-step process. And motor neuron disease might be a multi-step process. So if it is a multi-step process, then you would predict a particular pattern of increase of motor neuron disease with age. And I'm happy to go through the maths with anybody afterwards if they want. I've deliberately excluded it from here. But we could test whether this pattern exists using population registers. And if we test it, then it will tell us how many steps are involved if the relationship is seen. So the first thing we did was we looked in our Southeast ALS register, which covers the southeast of England. And the R-squared number there, the closer that is to 1, the more like a straight line the dots are. So an R-squared of 0.95 is very nice. It really tells us this does look like a straight line. So that tells us it's showing the pattern we expect if motor neuron disease is a multi-step process. So we then went to our colleagues in the Netherlands and said, you've got a population register, can we look at yours? They've got an R-squared of 0.99, it's about as good as you can get. In Italy, R-squared of 0.95. In Ireland, 0.99, and in Scotland, 0.97. So all of these different registers are showing the same relationship between the risk you get motor neuron disease and age. And if you combine them, then they show a very nice straight line, and that tells us they've all got really the same slope. And if we look at the slope, it's about 5. So that tells us that motor neuron disease is a 6-step process. So that doesn't mean it's 6 different risk factors, but it means that there's 6 molecular steps that lead between them to motor neuron disease. And you have to go through those 6 steps to develop the condition. And that would help explain these key features, because increasing risk with age, you can only see if you've had all of those 6 steps. So it takes time to accumulate the steps, so unless you've lived long enough, you're not going to accumulate them. There wouldn't be a single cause, because lots of different routes will lead to the 6 steps. Environmental causes would be difficult to find for the same reason, because, for example, smoking and radiation exposure both cause mutations. So there are 2 different things that can lead to the same molecular step. So you could easily imagine that, and with the different pathways. One gene variant could cause different diseases because, for example, if your steps were 1, 2, 3, 4, 5, 6, they might lead to motor neuron disease. If it was 1, 2, 3, 4, 5, 7, it might be frontotemporal dementia. So you can imagine that that could be explained. Reduced gene penetrance would be explained because if you don't go through all the steps, even if you carry a disease gene, you haven't gone through the other steps, so the disease won't be triggered. It would start in one region because those motor neurons would be the ones that have been exposed to all the 6 steps. And it would be specific to motor neurons because they're the ones for which those molecular steps matter, and the others wouldn't be. So in other words, our model of motor neuron disease risk would be more like this. You'd have whatever the genetic risk factors are at the time and the environmental factors all combine to make these 6 steps happen, and then you develop disease. So just to finish, genetics technology is mature and well-funded. We're beginning to understand the nature part of motor neuron disease, but the nurture part is much more difficult. The environmental studies are less well-funded. They're much more difficult to perform. So nature, nurture, age, and chance all play a part in motor neuron disease. And I'd just like to thank the funders who've also helped us make this work possible. Thank you very much.
Thank you, Amar. It's a wonderful sort of foray through motor neuron disease, where we are, where it's coming from, where it's headed. So we have time for a few questions, and I'm sure Mara will be happy to answer them.
Yeah. That GGCCC that's repeated a lot of times, is that happening in a non-coding region, or is that happening?
So I'll just repeat the question because you don't have a microphone. So the question was, the GGGCC region that's repeated, is that happening in a coding region? So that means a region that codes for protein, or in a non-coding region? The answer is it happens in a non-coding region. But we don't know why, if it happens in a non-coding region, that should matter. But it looks like it probably sucks up all of the RNA bits in the cell because it's a big repeated region. Normally, if you've only got a small region, the correct number of other components are there. But because it's so big, it sucks all of the bits up, and then they don't work properly. That seems to be one of the likely mechanisms.
So the exponential increase in the number of genes, so obviously that's got to stop somewhere.
Yes, it probably does have to stop somewhere. And I think if we can keep increasing the sample numbers, then it should keep going up to some extent, but until we've discovered all the genes. But really, well, it depends on what's, so the bigger you get, the larger the sample size needed to detect the gene, the smaller the effect of that gene. So at some point we will be detecting genes that have a very trivial effect, but they may still be telling us something about the pathway, and they may still be important for classifying the disease.
Yeah, John?
Terrific talk. Could you maybe give us a glimpse into the future of phenotyping and whether, for example, imaging, functional imaging, better clinical attributes or better classification of those?
Yeah, so that's a really important question. So how can we classify motor neuron disease in the future? There's lots of ways of trying to do it. So clinically, can we do it better? The answer is yes. In fact, I've got a paper coming out. I was just doing the proofs before this talk in Lancet Neurology, looking at how clinicians classify the disease, because it's very illogical at the moment, and that needs to be made better. But also we need things like biomarkers that might say, if you've got this particular cluster of chemicals in your blood, then that means you've got this type of motor neuron disease, and if you've got another one, you've got another type. I think the genetics will definitely help. For example, we can already say C9orf72 motor neuron disease might be different from other types. And the pathology might be able to help, but the real difficulty with the pathology is, although we know what it's like post-mortem and we can classify people then, that's no good in life, and we can't biopsy a brain or a spinal cord. So what we really need is some kind of imaging or other technique to show the pathology, and at the moment, those are in their infancy, but they are developing.
So you mentioned that it's a multi-step process. So does that mean, do you think that gene therapy has any hope for a cure after the disease has started, or that it could be used to prevent people who are susceptible?
So again, a very important question. The answer to that question depends on whether you think what perpetuates the disease once you've got it is the same as what's caused it. So it might be that what's caused it carries on causing it, and that's why it seems to spread. Or it may be that once it's triggered, you've got some kind of toxic event that happens in that set of neurons, and then that's transmitted through the network and causes this condition to continue. So if it's the second of those scenarios, then knowing what causes it might help you to understand the mechanism and therefore design a treatment, but it wouldn't necessarily lead you to the treatment. Whereas if it's the first of those mechanisms, it would. Whether we could use it to prevent the disease is much more difficult. to prevent a disease, you have to be able to screen the population for it. And if you screen for a rare disease, actually even quite a common disease, unless you've got an incredibly sensitive and specific test, you end up with lots and lots of false positives and lots of anxious people. So I think preventing motor neuron disease is going to be very difficult unless we can get a very, very specific test.
So how do you tackle with the flail arm patients or flail leg patients? Maybe they are the early stage of ALS when it's spreading.
So can I just make sure I understand your question? You're asking how do I tackle people who've got flail arm syndrome or flail leg syndrome clinically? Yeah. So if someone has flail arm syndrome or flail leg syndrome, firstly, they're more likely to be male. It's about a 9-to-1 ratio of male to female, which is very different from the rest of motor neuron disease. That's more like 3-to-2. And secondly, they're much more likely to have a very slow progression. It's not definitely a slow progression, but it seems to be. And one of the things that we don't really use in our classification, but which we probably should, is symmetry. And that's something that those two, flail arm and flail leg syndrome, which are basically very symmetrical conditions, they progress slowly and they're symmetrical, whereas most motor neuron disease is quite asymmetrical. It starts in one area, it spreads down, etc. And those people seem to have a worse outlook. So why symmetry should correspond with a better outlook or not is difficult to know.
Well, look, thank you, Amar, for a fantastic talk.
Okay.
So join me in thanking Amar and join us for a drink outside.